In this video, we are going to do an introduction to the specific defense mechanisms, also known as adaptive immunity. Now, this is going to be the branch or the part of your immune system that learns and remembers, giving you long-term protection against pathogens. So there's going to be lots of pieces to it. So this is the first of many videos. So specific immunity or acquired immunity is your body's targeted defense system. It's highly specific to particular antigens, those that are foreign substances that are going to trigger an immune response. So there's two main branches to specific or adaptive immunity. There's the humoral immunity, which involves B cells and the production of antibodies, and then there's cell-mediated immunity, which involves T cells that directly attacked infected or abnormal cells. There are specific videos for those two events that you should watch after you watch this one. However, in order for the specific immunity to respond correctly, it has to recognize the invader and decide which branch is it going to activate. And that's dependent upon where the antigen or where this pathogen comes from. So first, let's talk about antigens. An antigen is a substance that's going to provoke or elicit an immune response. Your immune system doesn't respond to the whole antigen. It's only going to be able to recognize a small piece of it, and that piece we call an epitope. Think of the epitope as like the face of the antigen. So if you have a whole body, it recognizes just the small piece or the face. That is what the epitope is. Now, the ability for that epitope to trigger immune response. So how great is that recognition? That's referred to as the antigenic determinant. The greater the recognition, the greater the response. Now, there are two types of antigens. An intracellular antigen, that's going to be like a virus or cancer cells that are going to be coming from within your own body cells, or there's going to be the extracellular, like the bacteria or the toxins that float around outside of your cells. So intracellular, those are going to be within the cell itself. Now, how it gets in there might be because it's a virus or a cancer. Extracellular, those are going to be found, the antigen is going to be found outside of the cell. Now, that's important because it's how or do you identify the antigen that determines on how we elicit the response. Now, not only do we need to understand that term antigen and that there's a couple of types, we need to talk about the antibody. So antibodies are also referred to as immunoglobulins. Those are proteins made by B cells in response to antigens. They are going to be what is going to be looking at that antigen specifically for that epitope we talked about, the face of the antigen, and it has to be recognizing that antigen in order to elicit that immune response, the correct immune response. So the receptor site on the antibody is going to have to recognize that particular site on the antigen, known as the epitope, kind of like a lock and key. Once they fit together perfectly and you turn it, then that's what's going to elicit an immune response. So looking here at this diagram for just a second, so let's just say this is our antigen. And notice here it has 4 different parts to it. Okay, each of those epitopes or faces can be recognized. Notice here that we have antibodies, and we have 4 different antibodies, because remember, an antibody is going to be specific for one epitope. So here the yellow antibody is specific for this basic triangular looking epitope. 4 different antibodies, 1 for each epitope. It is a specific, like lock and key, that it's going to recognize. This recognition is that antigenic determinant. The stronger the recognition, the stronger the response. So moving on here for just a moment in our terminology, in order for your immune system to be able to recognize and elicit that right response, it's going to have to be able to know and determine self versus non-self. So it's trained to recognize, your immune system is trained to recognize your own body cells, your own proteins as self. Anything else, like a bacteria, virus, or even transplanted tissue, can be seen as non-self and then will be treated as a threat. However, Sometimes your system can go haywire and your body will mistakenly identify your own protein or your own cell as foreign. And then that can lead your body to eliciting an immune response. And that is known as an autoimmune disease. So examples of that are going to be like rheumatoid arthritis, lupus, multiple sclerosis, Crohn's disease. And again, this is when something has gone wrong and your body is identifying self proteins or tissues as non-self. So then how do we recognize self? Because that's going to be due to markers that are carried out on your surface. So every cell in your body has surface markers, these proteins. These are ID tags that help with recognition and communication. So the most important of these are the MHC molecules, the major histocompatibility complex molecules, and they're playing an extreme role in the body identifying self versus non-self. There are three main classes of MHCs, and those are all going to be by their own gene. So let's look here first at the MHC class 1 genes. These are going to code for markers that appear on all nucleated cells, and they're going to help to present intracellular antigens to cytotoxic T cells. So remember, intracellular antigens are going to be those that come from within the cell. So it might be a virus, it might be cancer cells, and they're going to be displayed to cytotoxic T cells. MHC class 2 genes, those are going to code for responses that are going to be found for extracellular, and those are going to be for the markers that are going to be found on macrophages, dendritic cells, and B cells, and they're going to present that extracellular antigen to the helper T cells to elicit an immune response against extracellular antigens. So the two ones that we're going to talk about a lot are MHC class 1 for intracellular responses and MHC class 2 for extracellular responses, but there is an MHC class 3, and that's going to help elicit that complement system that we talked a little bit about in chapter 9. So if we have antigens and antibodies and MHCs, we keep talking about how they're going to present to one another. So then we have the what is known as antigen processing and presentation. So before your immune system can respond, it kind of needs that heads up, if you will, that, hey, we have something coming up. We have an invader. That's the job of these antigen presenting cells, or the APCs. Now, the examples of APCs are going to be macrophages, B cells, dendritic cells. But technically, as we just saw, all APCs all cells have the ability to be in APCs. Just the most common ones are going to be macrophages, B cells, and dendritic cells. So this is the stepwise presentation of antigen presenting cells. I'm going to run through it on a picture next, but also there is a video on this. So antigen presenting cells. So first what has to happen is an antigen has to come into the body. Now what we're representing here is an external antigen, okay, extracellular antigen. It gets brought into the cell, it's going to get processed through phagocytosis, etc. There's a video on that. And ultimately, though, the antigen piece is going to get attached to an MHC marker. Again, this one is representing from an external or extracellular antigen. If it was an intracellular antigen, the piece would be processed inside of the cell. But ultimately, in the APC process, you're taking a piece of antigen, you're binding it to an MHC marker, and then that marker with the antigen is going to be moved to the outside of that cell to act like a flag, if you will, to wave to the other body cells to tell them, hey, we have an immune reaction or immune response needed. So if it is an MHC class 2 with an extracellular antigen, as demonstrated here, then it's going to elicit that T helper cell response to fight off bacteria and toxins. If it is an intracellular antigen, so something that starts from within the cell, it's going to be presented on an MHC class 1 molecule, and it is going to elicit those cytotoxic T cells because it's going to be an intracellular. Now, there are other videos out there. There's one specifically on the APC process. Watch that. And then there's going to be videos on T cell eliciting an extracellular immune response, and then the B cells and cytotoxics for an intracellular. Watch those as well.